On a Saturday morning in 2009, researcher Michael Miller picked up a freshly caught salmon from his local supermarket. Miller and his colleague Craig Bennett work in the psychology department at UC Santa Barbara. Today, they are training one of their research assistants on how to properly perform functional magnetic resonance imaging, or fMRI, procedures using the dead salmon as a test subject. While the salmon was in the scanner, it was shown a series of photographs depicting people in a social situation with a specified emotional expression. The salmon was asked to determine what emotion the people in the photos must have been experiencing. Following the scan, the crew analyzed the data and found an anomaly. A section of the salmon's brain lit up in response to the images. Typical fMRI scans measure brain activity by detecting changes in blood flow. Yet, this fish has been dead for some time. Well, this wasn't just a training exercise. Bennett and Miller had a goal in mind. They wanted to illustrate how absurd, improper statistical approaches can lead to garbage results. In this case, Bennett and Miller used incorrect statistical approaches so you can actually see activity in a dead fish's brain. Their study warned about being intentional with your analysis and using the right methods for interpretation. But the safeguards that Bennett and Miller are highlighting here have been standard in the field for some time. So what is the real problem? This isn't an issue unless you're intentionally trying to be dishonest. This is a problem of reliability. Reliability refers to consistency. How likely are you to see the same results if you reran your experiment? Run the scan enough times, considering the large magnitude of collectible data, a false positive is bound to show. We would call this a type 1 error. Nothing is truly there, but the analysis incorrectly saw something it deemed significant. In the case of our fish friend, Bennett and Miller skewed the analysis to mark irrelevant noise from the scanner as significant brain activity. This is why proper methods are important and alludes to a growing issue in the scientific world. Psychology, neuroscience, and medical research studies have a huge replication crisis. Many experimental replications are failing, finding no statistical significance, calling concern to the reliability and validity of the original work. Honing in on the brain, Christiana Westland and Lisa Feldman Barrett, a leading researcher in the psychology of emotion, along with other colleagues, offer a revised way on how we should study brain-behavior relationships. They highlight three basic assumptions most traditional studies share that may be faulty: localization, one-to-one mapping, and independence. Localization is the assumption that a specific psychological event, such as speech comprehension, is caused by a single dedicated psychological process implemented in a dedicated neural ensemble. A neural ensemble can be defined as a brain region, circuit, network, or distributed pattern in the brain. One of the more known neural ensembles is Broca's area. According to localization, if someone has difficulty with speech production, it must be due to a specific impairment in Broca's area. One-to-one mapping is the belief that a dedicated neural ensemble maps uniquely to that psychological category. An example of one-to-one mapping is the belief that the amygdala is the brain's fear center. That is, it is uniquely and consistently being activated in response to fear, regardless of the situation, individual, or how fear is measured. The independence assumption states a stimulus will reliably evoke activity in a specific neural ensemble that produces an instance of the specific psychological category of interest, such as memory, executive functioning, attention, or vision. This neural ensemble is assumed to be studied separately from other specific signals that may moderate its function. Using a memory task as an example, participants are shown a list of words and asked to recall them later. Researchers focus on activity in the hippocampus, a region known to be involved in memory encoding. Under the independence assumption, the researchers assume that every time a participant successfully encodes a word into memory, a specific neural ensemble in the hippocampus becomes active, reliably signaling the formation of a memory, failing to consider the participant's level of attention, emotional state, or context. These early assumptions prioritize stripping away the noise and isolating features. Standard experimental designs for brain imaging studies typically sample relatively few trials per participant and use strict statistical thresholds to avoid type 1 errors. Consequently, studies generally observe and report only the strongest effects, producing islands of signal change that are interpreted as evidence for localized activity. These islands might actually result from studies that are insufficiently powered within individual participants, meaning there is a possibility to misrepresent the brain's true functional organization due to limited data per person. Strict statistical thresholds ignore weaker signals as noise, but studies specifically designed to examine those signals show that they are both reliable and psychologically meaningful. You can think of a mountain. The peaks are the most noteworthy feature, yet fail to encapsulate the full range of its features. If we treat the statistical thresholds like a mountain's tree line, of course the cleanest data will be at the peaks, but the valleys below are just as meaningful. Functional neuroimaging methods were initially assumed to reveal the unique neural locations for different mental phenomenon, like fear in the amygdala or cognition in the cortex. However, decades of study ironically produced evidence for the opposite. Localized neural computations contribute to a range of phenomenon. Let's use social cognition as an example. Given a mental task, like judging a person's beliefs, we can assume a specific brain region and a specific process. This approach evolved to multiple processes and multiple but still separate brain regions. And now, the current approach looks like this: interconnected networks and processes that all contribute to a single mental event. We went from separate faculties to distributed structures to brain function interfaces in a span of a couple decades. Our assumptions and methodology are shifting. The science behind the brain is increasingly considering the complexity it has to offer. Barrett's group suggests 3 new ways of thinking that match the complexity of researching brain behavior relationships. The first revised assumption is whole brain signals. It explains that a mental event, like memory or emotion, comprises distributed activity across the whole brain, not just isolated islands. Each sensory category has its own specialized region where sensory information is initially processed. For example, visual information is processed in the visual cortex, while tactile sensations are processed in the somatosensory cortex. However, in an interesting experiment to study the brain's multimodal signaling, researchers found that within these regions, there are unique patterns of neural activity associated with each type of sensory input. This means the visual cortex, traditionally associated with vision, can also respond to tactile or auditory stimuli. In The Brain That Changes Itself by Norman Doidge, neuroscientist Paul Bach-y-Rita says, "We see with our brains, not with our eyes." He uses the example of a blind man using a cane, sweeping it back and forth across the ground, feeding information to form a layout of his surroundings. Substituting his retina for the feeling of the cane, the man can still form sensations into pictures. These systems can compensate for each other because it's a feature of their circuitry. Many-to-one mapping refers to the occurrences of different processes or structures leading to the same result. This is also defined as degeneracy. We see this in our genetic code. Some of the amino acids can be encoded by more than one mRNA sequence. A mutation in the last position of the last nucleotide would not have an effect in coding glycine. There are also different biosynthetic pathways, each providing a different solution by which glycine can be made. In degenerate systems, many inputs can lead to the same output. This isn't to be confused with redundancy. For example, gene mutation studies showed that less than 30% of gonadotropin-releasing hormone neurons in the mouse hypothalamus are required for the onset of puberty. Here, structurally similar neurons carry out the same function, and the majority of them are labeled as redundant. In the human nervous system, pain perception is supported by multiple distinct pathways. One carries sharp, localized pain, like a pinprick, while the other carries more diffuse, emotional aspects of pain, like suffering or aching. Despite these tracks being anatomically different, damage to one of them does not necessarily abolish the perception of pain. The function is preserved or compensated through alternate routes. Using dice for example, if our main output is to roll an 8, in a degenerate system, we can roll a 2 and a 6, or a 3, then a 5, or 2 4s. We see that different elements result in the same output, versus redundancy, where we would roll a 2 and a 6, and then a 6 and a 2, same elements, same outcomes. Degeneracy also makes a system robust to disruptions. Imagine a parametric space consisting of 6 faces of a single dice. The goal is to combine up to 2 unique elements in the space to achieve the outcome. There are 3 different possible combinations of elements that can achieve this outcome: 1 and 6, 2 and 5, and 3 and 4. Now imagine that element 2 is eliminated from the space. Although one of the elements is gone, the system is still able to achieve its desired outcome. Complexity is correlated with degeneracy. If a parametric space consists of elements 3 and 4, given the small size of the space, only one solution is available to achieve the desired outcome of 7. Imagine adding more elements, like 2 and 5. Now 2 different solutions are available. Each can result in the same output. Adding 1 and 6 would make the system even more complex by increasing the size of the space. Now, at least 3 structurally different combinations, 3 degenerate solutions, are available that each can yield the same output. This idea is pronounced in our last assumption revision, the complex brain model. It describes how mental events emerge as a complex ensemble of weak, nonlinear interacting signals from all contexts: the brain, the body, and the outside world. In traditional neuroscience models, the anterior cingulate cortex, or ACC, might be labeled as the attention center or the error monitoring system. Under the independence assumption, we might study the ACC's role in attention by isolating tasks designed purely to measure attentional processes, ignoring memory, emotion, or bodily state. However, evidence contradicts this modular view. The same pattern of activity in the ACC has been shown to support different psychological function, such as attention or memory, depending on which other brain region it is functionally connected to at the time. For example, during the Stroop task, the ACC might be functionally coupled with prefrontal regions involved in cognitive control, supporting attention. In another context, during a memory recall task, the same ACC patterns may be part of a network involving the hippocampus, supporting memory retrieval. This means the function of the ACC activity is not inherent or independent, but emerges from its dynamic participation in different neural ensembles. Neurons don't have fixed functions. Their meaning depends on the active network and the broader neural context it's a part of. In the brain, when parts of a neural network are disrupted, a reconfiguration or compensation by other structurally different pathways can take place to counteract the disturbance and maintain function. We see in aging that the brain recruits more circuitry to perform the same task a younger individual might do with only one half of the brain. This is the reason some older individuals like to turn down the music when focusing on driving. Their executive function requires more bandwidth as they age. Degenerate complex systems are why it's so difficult to study disease or neurodevelopmental disorders in the human brain. Alzheimer's, Parkinson's, and autism all show significant biological variability and challenge a one-size-fits-all approach to classification. We cannot use traditional research methods to study degenerate systems as they often miss hidden compensation mechanisms. For example, in functional imaging studies, if 2 or more degenerate systems are capable of providing correct responses on a semantic task, none of the individual activated areas might be necessary in the first place. On the same semantic task, we see 2 distinctly different responses from different individuals. In lesion studies, we see lesions encompassing each of the areas activated in previous semantic tasks of normal individuals do not dramatically impair performance accuracy. So none of the cortical areas activated in the normal subjects appear necessary for correct performance on the semantic task. So what can we do? Degeneracy mandates the consideration of individual variability in research. A complete understanding requires systematically integrating data from both neuropsychological studies and functional imaging of healthy individuals and neurologically impaired patients. It suggests that multi-targeted drugs or neuromodulation targeting several elements simultaneously might be more effective in treating neurological disorders like epilepsy or chronic pain rather than single-target approaches. Ultimately, embracing degeneracy reveals a vast spectrum of possibilities and prompts introspection about current research methodologies in neuroscience. Considering the points made up until now, the human brain presents a puzzling paradox. Despite a fixed anatomy, characterized by its connectivity, its functional capabilities are vast, enabling action, perception, and cognition. This flexibility may stem from the brain's network architecture, which enables adaptability, efficiency, and resilience by organizing local interactions. Gerald Edelman was at the forefront of this concept. He introduced his idea of neural Darwinism, the theory of neuronal group selection, in 1978. He proposed that variation and selection within neural populations play key roles in the development and function of the brain, much like natural selection and evolution. It aims to bridge the gap in understanding the biological basis of psychological phenomenon by connecting it with developmental and evolutionary mechanisms. There are three core mechanisms central to Edelman's theory: developmental selection, experiential selection, and re-entry mapping. Let's examine these mechanisms in the frame of childhood. In early childhood, the brain, like an overgrown garden, is full of rapidly sprouting neural connections. In the earliest years, before and during the sensitive period, a time where children are more receptive to learning, around the ages of 2 to 6, genes act like a gardener, sowing seeds across the whole brain. These seeds grow into a wide variety of plants, some tall, some small. In terms of neurons, this means the brain is overproducing synapses in many areas. This overproduction ensures a rich and varied landscape of possible circuits, even though not all will survive. These initial connections are shaped primarily by genetic instruction and molecular gradients. They don't depend much on experience yet. For example, a child is born with the basic wiring for vision and language, even if they haven't yet seen words or heard speech clearly. Now imagine the child beginning to interact with the world. They hear language, see faces, touch objects, and explore everywhere. These repeated experiences act like sunshine and rain, strengthening some plants and leaving others to wither. If the child hears English every day, the circuits for English phonemes are reinforced. If they never hear tonal variations, like in Mandarin, the brain prunes those unused pathways. This is the process of neuronal pruning, where experience shapes the brain by reinforcing frequently used connections and weakening unused ones. As the brain matures, different parts of the garden start talking back and forth continuously. The visual, motor, and auditory systems send signals to one another in bidirectional loops. For example, when a child hears the word ball, and at the same time touches the ball, multiple brain areas coordinate their activity through re-entry signaling. This ongoing dynamic communication helps the brain integrate perception, memory, and action, creating a unified experience of the world. Edelman hated the notion that the brain had the same processing system as a computer. He did not like the idea was preset like an algorithm. But computers have changed, AI is everywhere, and rapidly changing how we interact with science in the brain. In 2024, about 1 in every 7 biomedical research papers published in PubMed found evidence of AI-generated text. 1 in 7 doesn't seem that bad until you realize that that is more than 200,000 papers in just 1 year. And that number is likely to increase exponentially as AI becomes more integrated into daily use. Large language models like ChatGPT are examples of foundational models, capable of taking information from large datasets, then applying that information to new tasks and new types of data, or what researchers called generalizing outside the training distribution. The problem is, if these training datasets are increasingly filled with information that already has traces of AI-generated text, this feedback loop over years and years can dilute truthful scientific information. There is a reality where the future, like a game of telephone, is a distortion to what is really true. We all recognize the rise of AI as a supercharged tool, but if we aren't careful with our intentions, deception and ignorance will and can creep into what we value as truth. Progress tends to move in the direction of righteous intention. We want to understand the brain, we want to answer the good questions, we want to solve the problem of suffering. Along the way, opportunity opens to capitalize on this goodness. For some, it's money, others, it's power. This is why feedback is so important. We need to step back from the forest to see the trees, and keep reminding ourselves of the direction we're going. We are all makers of our own path, and using our brains as an example, we should take time, sometimes doing things that make no sense at all, and be intentional about the way we view ourselves. And thanks for watching.